everyone! Thanks for choosing to watch our video. And in this video, myself and my team will be sharing our experiences of learning gene cloning and selection of recombinants as a story. So please keep watching. The story starts in the classroom of Shastra. The professor is about to start his lecture on ways to improve the copy number of GOI. Oh, but did I forget to introduce the lead of our story? Yeah, it's Paul, the smartest guy in the class. Oh my god, the bell rings and it's now time to conclude the class. The class disperses. But curious Paul is all excited to know more about cloning, but is also confused about where to start. But then, he remembers the basic components of cloning taught by his professor. So now, the first task of Paul is to isolate his donor DNA fragments. Paul chooses phosphoenol pyruvate carboxykinase to be his gene of interest. But what is phosphoenol pyruvate carboxykinase? Well, the mitochondrial membrane has no transporters for oxaloacetate. Therefore, before the export to the cytosol, the oxaloacetate formed from the pyruvate must be reduced to malate by the mitochondrial enzymes. The oxaloacetate is then converted to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase 1. The magnesium-dependent reaction requires GTP as a phosphoryl group donor for the reaction to happen. But why did Paul choose phosphoenol pyruvate carboxykinase to be his gene of interest? What significance does this gene have in our human body? Well, at low glucose level, it catalyzes the conversion of oxaloacetate to phosphoenol pyruvate that produces glucose from lactate and other precursors of the citric acid cycle. And at high glucose level, it catalyzes the conversion of phosphoenol pyruvate to oxaloacetate. It also acts as a regulator for the formation and the maintenance of memory CD8 plus T cells. On phosphorylation, at serenine 30 but 80 K1, there is a reduction of the binding affinity to oxaloacetate and promotes a serenine protein kinase activity, which helps in regulation of lipogenesis. So it's now time to extract our gene from the human cells. Human cells are broken by physical means by adding salt into the solution. The positively charged sodium ions in the salt help and protect the negatively charged phosphate groups that runs along the backbone of the DNA. A detergent is added and the detergent breaks down the lipid in the cell membrane and the nuclei. The DNA is released and these membranes are disrupted. To get a clean sample of the DNA, it is necessary to remove as much of cellular debris as possible, and often a protease is added to degrade the DNA-associated cellular proteins and other cellular proteins. Finally, ice-cold alcohol or ethanol is added to the DNA sample. The DNA is soluble in water but insoluble in the presence of salt and alcohol. By gently stirring the alcohol with a layer of a sterile lipid, a precipitate becomes visible and can be spooled out. The DNA sample can now be further purified and suspended in a slightly alkaline buffer and is now ready to use. Paul has now successfully isolated his donor DNA fragments, but he is also aware that he requires sufficient amount of his donor DNA fragments to continue the cloning process. For this purpose, he makes use of the PCR. The first step in PCR is the denaturation step. As in DNA replication, the two strands of the DNA double helix need to be separated, and the separation is done by raising the temperature of the mixture, which causes the double bond to break between the complementary strands, and this is continued by the annealing step. In annealing, the primer binds to the target DNA sequences and initiates polymerization, and this can only occur when the temperature of the solution has been lowered, and each primer binds to each strand of the DNA. Paul makes use of the primer designing tool available in the NCBI to design his own primer, and he chooses the first primer out of all. Since he knows an ideal primer should be of shorter length, as they are shorter, they will bind or anneal to the target more efficiently. It is also very important to choose a primer with high GC content, as G and C bases have stronger hydrogen bond, which will help in the stability. The primer should have an ideal temperature of about 50 to 60, and it's very important to check that your primer doesn't have dinucleotide repeats or more of one basis. Also make sure it doesn't form self-dimer or primer dimer. The third and the final step of PCR is the extension step. In extension step, new strands of DNA are made using the original strands as template. Taq polymerase enzyme joins the free DNA nucleotide together. The order in which the free nucleotides are added is determined by the sequence of the nucleotide that was existing in the DNA strand. The result of one cycle of PCR is two double-stranded sequences of target DNA, each containing one newly made strand and one original strand. The cycle is repeated usually 20 to 30 times, and by this way Paul gets his amplified GNA. Followed by the PCR, Paul now heads to blotting. Blot refers to the membrane on which a biological molecule like a protein or a nucleic acid are absorbed or immobilized. The process of transferring these molecules from gel to the membrane is known as blotting. When the macromolecule involved is DNA, the technique is known as Southern blotting. As we saw in the previous stages, the first stage is a DNA digestion, which is followed by the second stage, which is DNA gel electrophoresis. Followed by the DNA gel electrophoresis, the next step is blotting. Blotting means transferring the DNA fragments from a gel to a suitable membrane. The membrane can either be a nitrocellulose membrane or a nylon membrane. The nylon membrane is often used for its high tensile strength and better binding capacity. Blotting is done by transferring a suitable buffer in the tray. A support for the gel on the membrane is kept for the buffer. An absorbent paper or a blotting paper are placed on the support. They are placed in such a way that they imbibe the buffer and act as a weight. Next, the gel containing the DNA is placed on the top. A membrane of the same size as of the gel is placed over it. Again, a thick stack of blotting paper or absorbent paper is placed over the membrane. This is followed by 3 to 4 inches of paper towels, and this complete stack of gel membrane, blotting paper and paper towel are pressed by putting a weight on the top of it. The buffer or the liquid now rises through the gel, taking the DNA molecules with it. Once the DNA molecules reach the nylon paper, they become so tight to the membrane and the target. The remaining liquid passes through the paper and is absorbed by the paper towels placed at the top. During this transfer, the DNA fragments retain the same pattern of separation they had on the gel. Another transferring of DNA is done by electric field, which is known as electrophoretic transfer. This transfers negatively charged DNA molecules from the cell to the membrane instead of proteins. The next step is the hybridization or the washing step. In this step, the nylon membrane is now incubated with many copies of nucleic acid probes and labeled single-stranded DNA molecules under appropriate conditions. The probe will bind to the target sequences by complementary base pairing and form double-stranded DNA hybrids, so the probe hybridizes with the DNA fragments that is complementary to it. The unbound probes are removed by washing. The next step is the detection step. The detection of unbound probe is carried out. The localization of the double-stranded DNA hybrid was found in the previous step. The probe is detected by autoradiography, fluorescence, or color change depending upon the label used in the probe B. After successful isolation of his gene of interest, Paul had to now find a suitable vector for his cloning. He remembers his teacher explaining that the vector is a small circular DNA molecule which is capable of reproducing independently of the host. From what he learned in the class, Paul now goes about to select PKIL18 as his vector. PKIL18 are derivatives of PUC18 plasmids. In PUC18 plasmid, the CDB killer gene has been fused in the phase downstream from LACP MCS18 multiple cloning site. But why did Paul choose PKIL18 vector? These plasmids are capable of producing high copy number, and the plasmids have similar restriction site to that of his GHOI. The significance of choosing this is that they possess a CDB gene, a potent poison of gyrase, which can later be exploited for the selection of positive clones. The third and the most important step of any gene cloning process is the manipulation of a gene of interest in our vector. Much like the discovery of an enzyme that cut DNA, the discovery of an enzyme that could join DNA was preceded by earlier salient observations. In 1972, Berg separately cut and ligated a piece of lambda bacteriophage DNA or E. coli galactose operon with SV40 DNA to create the first recombinant DNA molecule. These studies point out that the concept, because of the universal nature of DNA from any species could be joined together. In our case, SAC1 and SAC2 are the restriction site. GOI is cleared as mentioned in the above slide and manipulated. Here, ampicillin acts as a selectable marker. Since our plasmid possesses only one of the restriction site, other restriction site is added as adaptive molecule. Adapters are a pair of short nucleotides that anneal together to produce a short double-strand DNA fragment containing different sticky ends. Then the fragments produced are attached to the plasmid with the help of a DNA ligase enzyme. Followed by the manipulation, the next step after manipulation is the transformation. Transformation is the ability to transform bacteria to induce the uptake, incorporation, and the expression of foreign gene material. This was first demonstrated by Griffith when he transformed the non-lethal strain of bacteria into a lethal strain by mixing non-lethal strain with 8 inactivated lethal bacteria. Typically, transformation is performed to increase the competence of the cell in a GOI. The transformation increases with low DNA level. As we know, there are both physical and chemical methods, but here we focus on the electroporation, which is a physical method. The term electroporation was coined by Human and co-workers in 1982. Using electric current, any biological molecule such as nucleic acid, drug, chemical, or a viral DNA can be inserted into a live cell. The principle is that the eukaryotic cell membrane or cell wall is mostly made up of lipids, phospholipids, protein, chitin, pectin mostly. Once we apply current to cell suspension, some pores are created in the cell membrane. A current of 1 to 1.5 kV can be applied only for some milliseconds for effective insertion, and cells can be competed. The competed cells are now ready for the intake of foreign materials. Here, for instance, a DNA. We will discuss the entire process present method using the example of a gene transfer. It is also known as pulse generator 2. In the very first step, the plasmid is constructed for transformation. A gene of interest along with the marker DNA sequence ligated into the plasmid. The selection of a plasmid depends on the type and the size of the nucleic acid used for the gene therapy. A population of target cell is taken into the suspension filled with the electroporation buffer. The electroporation buffer protects the cell suspension from mechanical damage. Along with it, the target DNA, plasmid DNA, is taken into the curvet. Then it is placed in an electroporator. The electroporation machine or the device is known as electroporation machine. On either side of the curvature, 2 aluminum electrodes are fitted. The device is connected with the power source. When it switches on, the circuit completes. The electrical pulses pass through the cell suspension. This will create temporary pores in the cell membrane. Notably, the entire process completes within seconds. Changed molecules like DNA can pass through the pores into the cytoplasm of the cell under the influence of current. After completion of the entire process, the viability of the cells is checked. The final step, the transformed cells are cultured. But why did Paul choose electroporation for his transformation? Here are 4 advantages of electroporation. One of the major advantages of the present method is high effectiveness and rapid transfection rate. Electroporation can transfer foreign materials effectively faster than any other viral or non-viral vectors mediated delivery system. In comparison with the viral vectors, it's safer. Also, it's non-toxic, non-allergic, and non-mutagenic. After transformation, Paul performs selection to ensure the successful cloning. The 2 possible ways of selection are selection based on the plasmid character and selection based on the insertional sequence. For his first level of selection, Paul chooses a selection based on the plasmid character. The recombinant plasmids are transformed into a ampicillin-sensitive, CCDB negative and CCDA negative host cell. Because our plasmids are ampicillin resistant and contain the CCDB toxin, for which CCDA can counteract as antitoxin. The host is cultivated in a minimal media with ampicillin antibiotic to ensure the growth of the desired cells. Transformants are the cells that have uptaken the foreign DNA by transformation, whereas non-transformants are the cells that have not undergone transformation. Ampicillin acts as an irreversible inhibitor here. The beta-lactam ring binds the transpeptidase active site of the enzyme transpeptidase, which is needed by the bacteria to make the cell wall. It inhibits the third and the final stage of bacterial cell wall synthesis in binary fission, which ultimately leads to the cell lysis. When ampicillin-resistant gene is present, it directs the production of the enzyme beta-lactamase that cleaves the beta-lactam ring by hydrolysis, which turns the ampicillin into an inactive metabolite and the bacteria are able to survive. Non-transformants are sensitive to ampicillin and cannot survive in an agar plate which contains the antibiotic ampicillin, whereas transformants are resistant to ampicillin and can still survive in an antibiotic medium. For the second level of selection, the host containing the non-recombinant plasmid, the CCDB gene gets expressed via transcription into mRNA, the translation into amino acid. The amino acid peptide gets folded into the CCDB protein, which is a cell-killing protein and is a potent poison of the bacterial gyres. It is also an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of double-stranded closed circular DNA. Inactivation of DNA guides. It is known to act on DNA guides in two different ways. The CCDB poisons the guides DNA complex by blocking the passage of polymerase and leading to the double-strand breakage of DNA. In cells that overexpress the CCDB, the subunit A of DNA guides has been found as an inactive complex with CCDB, which finally leads to the death of the cell. In the host cell containing the recombinant plasmid, the CCDB gene will be insertionally inactivated by GOI, so that the gene can't express and produce a functional protein, which leads to the cell survival. Thus, the cell growing on an agar plates indicate that they contain the recombinant plasmid, in which the CCDB gene has been insertionally inactivated. We can go further by constructing the master plate, which contains only recombinants. So everyone, this is how Paul successfully increased the copy number of his GOI, which is the phosphoenol pyruvate carboxykinase. Thanks for watching, and we hope you liked the video. And here are our teammates who worked really hard to bring this video to you. So please drop in your comments. Thank you.